we're talking about to differentiate between variant level evidence and experimental evidence, and make sure that if you're going to upgrade one piece, that you're not essentially double dipping and upgrading both the variant score and the experimental evidence score for the same piece of evidence. I know particularly we've come across a lot of genes that are moderate or limited, and that could easily inflate, particularly if they're on the edge of a category, and bump it to the next category. So I think that's something important to consider too.
All right. So what would you think about this situation, Marina, where they had, you know, they had basically done a bunch of experiments all showing that, I guess the protein isn't trafficked correctly, so it doesn't get to the it doesn't get to the plasma membrane where it can then perform its normal function. So would you agree? I mean, is this the kind of, is this how you would score this evidence, or do you feel like?
Yeah, I agree. I think that this is an appropriate separation of variant level and experimental evidence. I think in general we're pretty conservative in the hearing loss group with upgrading variant points. We often do just give the suggested number of points, but I feel like these types of evidence are completely separate. I could see if you took the third experiment in red and used that to upgrade the variant points and the experimental evidence points, that would be double dipping. But I think the way you have it split is
I agree with that, Marina. I typically, a lot of times I'll usually only see, like, maybe they looked at the expression with the variant, and in that case I would give it the default 0.5. I would use that to give the default 0.5. But I thought in this case, since they did not only expression, but they also looked at the processing and the localization of the protein, in this case I might consider giving it like 1 point or something like that, but then and still not double counting when I use the third piece as experimental evidence.
Yeah, I agree. This is a pretty thorough workup of a particular variant.
So what I'm starting to actually recognize is a pattern here with some of these things that we've looked at in terms of carrying it for variant level or experimental evidence. This is Bryce Seifert talking, by the way. And what it seems to be is when it generally relates to protein function in terms of trafficking to particular parts of the cell, expression, or basic like metabolics of the protein in terms of does it get processed correctly, does it get to the right part of the cell, that seems to be counted more often with variant level evidence. But when it disrupts its function within a particular pathway or I guess cellular compartment, in the sense that if it were some sort of a channel or pore that needs to release calcium, and then without that it results in an organismal or pathway defect, that seems to count more towards experimental evidence. And so it may require further discussion, but I'm wondering if we want to, from these kinds of discussions and looking at these types of examples, would we want to consider updating the SOP in terms of the way that we look at experimental evidence and variant level evidence that relates to experimental data, and get a little more granular with it in terms of, okay, if it messes up a pathway that a protein's involved in relation to the particular disease, in this case oligodontia, then it could be counted as experimental. And if it relates more to just a general function of a protein that most proteins would need in the sense of localization or expression or processing, does it need to be variant level? I realize that can get sticky in the case of maybe like collagens or something, but I don't know, maybe something to think about.
Yeah, and I agree, Bryce, and you know, and I think you're right too. I think it can be, these situations can get sticky sometimes. Sometimes it's not really exactly clear. And one thing that Jonathan had mentioned to me was to really look at, if you are not sure where to count that evidence, try it in both places and see how it affects the points, and then discuss with the expert as to, you know, does it affect the classification in any way? And also discussing with the expert as to whether they feel it's more variant versus experimental evidence, because it can be tricky. But I think you know, my understanding is that you have it exactly right. Like if you can link this functional alteration to the known disease mechanism of the disease, then that would count more towards experimental. And I know Courteny has some more examples as well of where she had kind of parsed this out. But does that make sense to everybody? And we can, I mean, one of the things that I'm trying to do as well is just to collect a bunch of examples for case level evidence and experimental evidence and put all that together so that that will at some point be available for everybody. So I'm working on that at the moment, and we'll have, you know, the gene curation small group review that, and Jonathan as well, just to make sure that everyone is in agreement. So hopefully we'll have that as a resource in the future as well. So, all right, thank you, Jen. So the next example is from Courteny.
Yes, hi everyone. So I did like Bryce's point, but I do want to point out that this may be an example where it could be a little tricky between variant and scoring experimental. What I was doing here was looking at fibrillin-1 and Marfan syndrome. And so we know that mutations in fibrillin-1 can cause this syndrome. And what this particular experiment did was go through induced pluripotent stem cells that were isolated from patients that were harboring these pathogenic mutations. And they were differentiated into smooth muscle cells, and then the deposition of fibrillin-1 into the ECM was measured. And so contractility of these muscle cells in response to carbachol stimulation was measured, which you'll see on the very far right. And I counted this towards experimental evidence. You could look on the left-hand side and look just at the deposition of fibrillin-1, and again, it would kind of look like you would look in any other variant evidence like, oh, there's mislocalization or there's less deposition. The reason why I still supported this for experimental is because lack of deposition of this extracellular matrix is what caused the thoracic aneurysm and aortic dissection phenotype in Marfan syndrome. So I felt that that was really showing me why this was happening, because there was less collagen. And then further, because these cells were now differentiated into smooth cells and looked at the contraction of them, we can see that there's loss of contraction, which is the red line on the very far left diagram there. And so for me, I counted this towards functional alteration patient cells because the destabilization of ECM was the hallmark of this aneurysm, and it supported a disease mechanism, not just a variant level evidence.
Great.
Okay, if you want to go to the next one, Jenny.
Sure. Or does anyone have any comments or questions about that one?
This is Anna. It's not a question pertaining to this case in particular, but I'm trying to make sense of all of this, which is really helpful. And the only question I have is, I thought that when we curated variants, we would always be aiming at establishing whether the functional evidence, whatever it may be, addressed the disease mechanism. And I feel like only the experimental data would be targeting that, and that the variant level data as discussed here, metabolic or biochemical processing, may not necessarily target the disease mechanism.
So you mean in this, in general, or so not this particular case?
No,
so
yeah, in general, am I correct in understanding that there's sort of a line between experimental data being the type of data that targets disease mechanism, and I'm just trying to contrast it with my prior understanding, which was any functional data for any variant that you are adjudicating should be actually addressing or targeting the disease mechanism, and that if the functional study doesn't address the disease mechanism, perhaps it's not a strong functional study.
Right. I think I see what you're getting at. So I mean, I think when we're thinking about things that count towards the variant level evidence, I mean, you know, a great example of that, as I said at the beginning, is the you know, splicing assay. So it tells you that this gene is the mRNA isn't. The RNA rather isn't correctly spliced, but it doesn't really tell you anything about the disease mechanism, but it is telling you that there's something wrong with the way that this gene is being expressed. Whereas in this case, if you have evidence that is linking that particular variant to the disease process, then we felt that that's also implicating the gene in the disease as well, because when you have a variant in that gene and you can then link the effect of that to the disease, then you can count it in experimental. Is that making sense? I feel like I'm not explaining that.
I'm following you. Am I correct then in my understanding that then in this framework, the experimental data is stronger than the variant level data?
Well, the variant level data, I mean, you can get up to 12 points on that, whereas the experimental goes up to 6. So, I mean, you know, it's weighted in that way.
Well, I feel like this is kind of like a nuance of the variant level data, because when we say variant level data, we're actually meaning like human genetic evidence, and this is kind of a parentheses in that you want to be sure that the variants are convincing enough to link human cases with the gene with the disease. So, you know, I think that they both have different purposes in this case. And we've definitely come across limited genes that are mostly human cases or limited genes that are mostly experimental evidence. And you can't necessarily weight one more versus the other, but they're just different, I would say.
Okay. Thank you.
Okay. All right. So the next example, again from Courteny.
Right, so I was looking at the gene FHL1, which is 4.5 limb domains. It is mutations in this X-linked gene cause Emery-Dreifuss muscular dystrophy with HCM. And so they expressed these FHL1 mutations that were linking to HCM into the C2, C12 myoblasts, and they showed reduced expression and degradation by the ubiquitin proteasome system, and that's going to be the top column of B. They also transfected these mutants and showed reduced expression by immunostaining. So I counted this towards variant evidence because it shows that there's instability within cells and represents a loss of function mutation, but they didn't really tell me anything about the disease mechanism, and since the UPS isn't implicated in HCM or in Emery-Dreifuss, I wouldn't count this towards experimental evidence. And if you want to go to the next slide.
Okay.
If there's no questions.
Any questions about this one? All right.
Okay. So continuing with FHL1, this paper then went further to take neonatal murine cardiomyocytes, and they transduce them with virus with the same mutant constructs. And they show that there was not only mislocalization of FHL1 and incorporation into the sarcomere, but they went a step further and showed MYBPC3 localization and incorporation was perturbed. And MYBPC3 is one of the genes that is involved with 40% of hypertrophic cardiomyopathy, and so is disruption of the sarcomeric organization. And so because there was a change in a known gene associated with the disease, hypertrophic cardiomyopathy, as well as a disorganization of the sarcomere, which is what holds or stabilizes the heart, then I counted this towards experimental evidence because this was supporting disease mechanism. Any questions? Okay, you can go to the next one. And then they had another, again, with the FHL1. This was pretty neat. They virally transduced rat-engineered heart tissue with the mutations, and they measured the ability of this engineered heart tissue to contract, and they showed that there were changes in the contraction parameters. So this, again, was showing and supporting the disease mechanism that once you expressed these mutant FHL1 isoforms, that you were no longer able to contract normally as wild type was. And so this, again, was experimental and put in model rescue cell culture model system.
Any questions? Okay. So, so moving on, I think on the last call we had discussed several examples for autosomal dominant disorders, and we had started talking about autosomal recessive, but I thought we could start at the beginning with the autosomal recessive disorders just to review. And so this was a slide from Marina. So Marina, do you want to talk about this one?
Yeah, sure. We often work with autosomal recessive diseases, so we think about this quite often. But a little bit of text in the SOP that could easily be missed is in the variant scoring to say that 2 variants are in trans when you're scoring for autosomal recessive. And so I just wanted to point out again that if you have a homozygous case where a person has 2 of the same variant, you don't need to confirm, and that would be on the left of the pedigree that you see. Whereas if you have a compound het where they receive 1 variant from each parent, you really do need to confirm, at least that they've done sequencing of parents for a pedigree, that the variants were indeed in trans. And this is illustrated by the example I have on the next slide. So
can I ask a question real quick on your last slide?
Yeah.
Do they need to confirm that it's would you want them to confirm so that you know it's your missense variant and not a deletion? It's for your homozygous?
Oh, for the right. So that's a question I've had too, Christy, because, yeah, you could have a large deletion on the other allele that was just not picked up by sequencing.
It's true. And the example that I have on the next
you could also just have a run of homozygosity, right? It doesn't have to, it could be non-copy number.
Yeah, it could be UPD, that's what I was going to say.
Yeah, UPD or, I mean, since they're consanguineous in your pedigree, I mean, it makes sense.
Right, and you have
in this instance, you wouldn't need it, but if you didn't have known consanguinity, I don't know.
I think it's a great question, and I mean here with 2 affected SIBs, it would be, I guess, unlikely to be something like UPD, but yeah, Christy, I see what you're saying, that it could be, yeah, a large deletion on one allele and the variant on the other, which would still implicate the gene.
Probably, if your gene can be caused by truncating variant.
Right, right. Although if both copies are affected in some way, to a large deletion on one and a missense on the other.
Yes, or there could be the strange situation, like I have on the next slide, that it's actually a haplotype, and you wouldn't necessarily know. So in this case, we came across a consanguineous family from Turkey, and they had profound hearing loss, and they had 2 variants, but the variants were actually in cysts. And so if you didn't have this confirmation, you might be potentially confused, not treat them as a haplotype, and have a lot of non-segregation because you need essentially homozygous double copies. The people who only had 1 set of the haplotype did not have hearing loss in this case.
Right. Yeah, so this is a really nice example for the confirming that these are in trans when you see 2 variants in a patient. It's a really nice example.
Yeah.
Okay. All right. And Marina, this was your example too?
Yep. So a lot of the examples I have, we've kind of already discussed them, but so this is another autosomal recessive example for Usher Syndrome CDH23. The missense variant that I have circled at the top, the only evidence that the authors in the paper have is in silico predictions. So they've shown that it's in a highly conserved region of CDH23. It's actually located in the extracellular domain, and these are related to cell-cell adhesion. So it is a highly conserved domain to allow for the assembly of the dimers. However, there's no further information, so we would recommend to downgrade this missense variant in this case if you were going to score it at all.
Can I ask you, Marina, when you downgrade, I mean, how would you typically score something like this? Because, you know, we come across this issue a lot too, and
yeah. So to be honest, we're usually pretty conservative with our variant scoring, and instead of downgrading, we would usually, if missense variants don't have any functional evidence supporting, particularly because hearing loss, there can be multiple inheritance patterns for one gene, autosomal dominant and autosomal recessive, depending on the variant type. If there's no functional evidence for a missense variant, we often do not score it.
Oh, right. Because we've had this discussion a lot with our HDM group, as Anna will tell you as well, and we've been giving them 0.1 just to recognize that they're there, but just giving them a tiny, you know, because it's like a variant of unknown significance. I mean, unless it's too common. So that's how we've dealt with it, and it really, you know, I guess overall doesn't really affect the classification, but that's kind of how we've handled that.
Yeah, I feel like that's pretty similar. 0.1 points versus 0 points isn't too much, and
it's
we
tiny.
often see a lot
right.
of missense variants, so I feel like it would start to add up pretty quickly for us if we counted all of them. But I think that's a pretty similar approach.
Right, and we'd actually taken a look at this to see whether giving these variants 0.1 would actually change classifications, and you know it didn't. For most of these genes, they had so few missense variants reported anyway that it wasn't really affecting it. So. All right, so this is an example that I had looked at, and this is the PDE10A gene, and there was a homozygous missense change found in patients with a hyperkinetic movement disorder with onset in infancy. So this is the family here, so kind of an interesting family history. There are 4 affected siblings, and the father is also affected and has an affected sibling. This family was analyzed by West, and for all the individuals with the little star, they had genetic testing performed. So it is a homozygous missense change. It was rare in XDAC, and there were no homozygotes in XDAC. This tyrosine at 107 is highly conserved, and based on in silico predictions, it was probably damaging by polyphen, deleterious by SIFT. And of interest, this gene is intolerant of missense variation, so that's already, telling you you might be suspicious of this particular variant. So they went on and did some experimental work to further look at the impact of this variant, and what they did was to express the variant in HEK293 cells. So this shows their Western blot, and this is the lane for the variant that we're interested in. As you can see, this upper band, I think that's a non-specific band in this Western. They don't mention this in the paper, but it's kind of equal intensity in all of the samples. But you can see for the cells expressing the variant that we're interested in, the level of protein is a lot less than in the wild type. So definitely reduced expression of this variant in cells. And then they had gone on and looked at the level of expression relative to GAPDH as a control, and you can see here this is the variant that we're interested in, and this is the wild type. And there was another missense variant also identified in this paper that, as you can see, has reduced expression. So that was one piece of evidence. So I would certainly count that as supporting evidence towards the variant, and because this is a missense, I probably would have given it default. But then they had also gone on and done PET analysis to look at the level of PDE10A in the basal ganglia of one affected family member, and showed that there was a 70% reduction in all areas of the basal ganglia. So I thought that was really interesting, especially considering that this is a hyperkinetic movement disorder. And actually, I should also mention this finding was reported in the American Journal of Human Genetics, and in that same issue, there was another paper reporting de novo variation in the same gene in patients who had definitely a similar phenotype. I mean, there were some differences too, and it actually has a different MIM number in OMIM, so I think they're seen as different disorders and different inheritance pattern, but they had a childhood onset chorea. So I thought that was kind of interesting. So this is one of these situations where I have not discussed this with an expert. So they're basically showing reduction of the protein in an area of the brain that's known to be involved in movement. So I kind of feel like this is one of these, it's not necessarily linked to the disease mechanism, but it kind of is because it's showing a change in an area of the brain that is known to be important. So I think this is something that I may discuss. Would I count this towards experimental, or would I count this towards case level evidence? So does anyone have any thoughts on that?
I feel like I would maybe count this towards that strange subsection of expression where it's decreased in patients.
Yeah, and that was kind of what I was leaning to as well. And I think just because it is in that particular area of the brain, yeah. So I would probably give a default for the variant based on the Western analysis, and then include this in experimental.
I would just worry, though, about putting it into expression because it's from an affected individual in that family with the PDE10A mutation, which we can see already has reduced expression. Now, if you could show PDE10A is reduced in someone else with a hyperkinetic movement disorder in a different mutation, isn't that how you would normally count for expression?
No, you'd probably count by the fact that the controls in that graph they see there, Courtney, on the right, the bottom right, where it says caudate and the putamen, if those controls are wild-type controls, that's usually how I think we've counted in the past in terms of just general expression.
Well, no, see, that's where I think with expression there's been a misunderstanding that if there's reduced expression in patients, you count it, but it's not in the patient with the mutation in that gene. It's saying, for instance, if I look at 500 individuals with hypertrophic cardiomyopathy and they have all differing genetic reasons to cause HCM, but I see there's a change in MYBPC3 levels, which is linked to HCM, then I count that for expression because overall we're seeing an increase in this gene or this protein product in individuals with the underlying disease.
Yeah, I agree. It's because it doesn't say that it's this is the effect of the mutation. That's why it's the transcript is going down, or is it like, you know, yeah, you need a proper control to make an inference, like, you know, like Courteny said, you need that another hyperkinetic thing where, you know, that this doesn't go away because of an effect of the, you know, phenotype in this patient. This PD10A could go down as well.
Right.
You can rule that out.
Right.
So
it's like if you have a null variant and then you get NMD, and that's why you don't see expression. It's understandable. Thanks.
Yeah. I mean, I think I would be more inclined to almost count this still towards variant because you're just showing that there's reduction of this protein within a region that is known to be important for the disease. I think if you, go ahead, sorry.
No, that was my struggle. I mean, I think, you know, basal ganglia are, you know, obviously this is an important region of the brain in movement. So that was why I was kind of tending towards experimental, even though it is a, you know, patient level, it's patient with a variant, and we're looking at expression. So,
yeah,
I still think,
borderline ones. I would just say, obviously, we wouldn't want to upgrade the variant points and count it for experimental.
Yes. Absolutely, and that, you know, that may be a situation again where, you know, you can look at it in either place and see is it gonna impact the classification at all. Talk with the expert and you know, decide what to do. So
and the thing is, Jenny, with this, I may just be misunderstanding, but the fact that they're seeing PDE10A expression in general in the brain, and that's the tissue that's affected that results in the movement disorder, I could understand counting that for expression changes, or not expression changes, but expression in general in the tissue of interest.
Oh, yes, I do agree with that, Bryce. The control alone showing the expression in the basal ganglia, I think, but I don't think you could use the inference that it's reduced. I think the reduced in the patient should go towards the variant evidence for that person. But the control, I agree.
Right, I completely agree with that. If we were saying, oh, it's experimental evidence where there's reduced expression in the patient with the disorder, I agree that it couldn't be due to a variant in that particular gene. It would have to be a patient that has a variant like that in another gene or whatever. No. I understand. Okay, thanks.
Also, it's just N equals 1, so there's that thing too.
Right, exactly. It's only one case. So, yeah, we've had 10 patients from different families. You know, obviously that's going to increase the evidence. So thank you. I think this is really helpful discussion to get out there, so thank you. Okay, so Marina, this was another of your examples.
Oh, yeah. So this is similar also to what we've been discussing. So this is another Usher Syndrome variant in USH2A, which is Autosomal Recessive, and it is a splice site variant. It's confirmed in trans. The patient has 2 predicted or proven null variants in trans. In silico prediction tools, you predict the variant splicing, but then there was additionally a mini-gene splicing assay to confirm the skipping of exon 26. So I think in this case, that would be variant-level evidence. No disease mechanism suggested at all. If you would upgrade the points, you could probably do it to the variant.
Great. All right, any questions about that one? All right. Oh, so this is another example that I had looked at. So this is the hyroxy1 gene, which was found to be altered in patients with an early onset myopathy. In this particular family, there were variants that were found in trans. So important to know that the mother was a carrier of this missense change and the father was a carrier of a splice site change. So they had gone on to do further analysis of the splice site change. It is a +1, so you would assume that splicing would be affected. But they had looked at cDNA and found that in the affected individuals from this family, you're seeing 2 bands. So one representing the allele that has the missense variant or the nucleotide change causing the missense variant, and the other representing an abnormally spliced product. And they were able to sequence that and find that the splicing change had caused skipping of exon 3. And when they sequenced the upper band here, they found that all of the transcripts basically had the nucleotide change responsible for this missense alteration. So they felt that this splicing change basically was completely abolishing normal splicing in this case. So some nice experimental evidence to show that this splicing variation is indeed affecting splicing. And when you look at where this lies within the protein, so as I mentioned, the splice change causes deletion of exon 3, and that region typically would code for the pyridine nucleotide oxyreductase domain of this protein, which is an important domain in its function. So you get a big deletion there, again suggesting that this splice change would impact the function of the protein. So they then went on to look at impact of the missense change on the protein. And I should mention here that this particular protein, so it's an oxidoreductase. What they had done was to look at yeast complementation assays. So these yeast are missing a glutathione reductase gene and are then very sensitive to hydrogen hydrogen peroxide. So what they had done is then done a yeast complementation assay. So they had put in wild type and also the variant with this particular variant, and shown that this variant does not rescue the oxidoreductase activity in these yeast. So here's our variant here, and this is with the hydrogen peroxide. You can see that they are sensitive and it's not rescuing. So that was certainly good evidence that this variant is impacting the function of the protein. And then it also gone on to look at the expression of this protein in the muscle of these family members. So here you can see family A. These are the folks that have these two variants. This is the protein that we're interested in. This is a non-specific band up here, but you can see that there's reduced level of the protein in these patients. So this is in skin fibroblasts and also in skeletal muscle as well. So overall, I mean, I guess in a way, yeah, this is a similar situation to the level of the PDE10A in the basal ganglia. Here you have reduction of the protein in the skeletal muscle, and in this case, yeah, I would have counted this towards the variants, I think. So you're just seeing reduced expression of the protein. But for both variants here, there really is, you know, quite a lot of evidence. We see evidence for impact of the splicing variant, and then how that would affect a critical domain of the protein, and then you have various evidence for the missense as well. It's predicted to be pathogenic. It's shown to be impacting function on a yeast complementation assay, and then you're missing or reducing the amount of protein in these patients. So I would definitely upgrade this in this case. I mean, definitely I think 2.5, maybe higher. Yeah, there was a lot of evidence for this one. So any thoughts about this one?
So do you also consider these evidences in the experimental section, like the models and rescues?
No. I would count it only once. So I think that's what, you know, as Marina had mentioned, you don't want to double count. So I'm counting this towards the variant, but
yeah, that's what I was just wondering whether this qualifies as as a model system, and there is also a rescue because complementation matters. So and it's an enzyme which is conserved, so I don't know.
This is Courtney. We've had this before with yeast, and typically, from what my understanding was looking at what yeast complementation assays were, they were really just trying to show that the gene that they put in or any mutation was just affecting a pathway in the yeast. And so while you could potentially have a rescue, it's just showing that it's within a pathway. And so not necessarily a rescue of any function for the disease or the disease mechanism. So I think we've had one of these before where we did count it more towards variant just to show that there is no ability for this protein to function. But I mean, I can understand how it kind of sees what it looks like a rescue, but I think unless you really know that the disease mechanism for this specific disorder has to do with this oxidant, you know, reductase and oxidative stress, I don't think you could count it towards the rescue, because that would definitely inflate your points up to like 2 to 3 if you cut that out.
Yeah, I think there are definitely some aspects in yeast. We came across an example when we were working on the gene curation paper, and I had presented a couple of the curations for expert review, and in one case there was a yeast model, and it did show, I think it was alteration of vacuole sorting. And the expert had said, this is actually a common model that we use for this disease area, and I would count this as an animal model or as a model of the disease. So I feel like in that case, it's a little different. So I agree with you, Courteny.
Right. So that's a really good point. You know, just what are these assays showing? And this is where the experts can come in, just, you know, with all this great advice as to where to count this evidence. But I think in this case, because there wasn't really a lot of information to tie this particular activity to the disease process, I decided to count it towards variant. But, you know, I think this just illustrates that there is, you know, there can be some decision-making to be done, and, you know, really sort of thinking about what these experiments are showing you in terms of where to count the evidence. So let's see, we have, I think, a couple of minutes more. I can race through this one, and then we just have a couple of minutes to go. So, so this is for Pompe disease and the GAA gene, which is the cause of Pompe. Pompe is also known as glycogen storage disease type 2. So these patients have accumulation of glycogen in their muscle tissue, and it's a huge range of phenotypic severity with the infantile form causing early death in patients less than 1 year of age, and then all the way up to adult onset forms as well. So this was another situation where I felt like there was actually a lot of evidence, and I actually particularly chose this case when I did the curation because both of these variants are well-known pathogenic variants within the GAA gene for Pompe disease. So the variants were shown to be in trans. So from the mother, the patient had inherited this splice site variant, which has been studied extensively and is actually shown to basically be a sort of leaky splice site variant. So it causes incorrect splicing in about 80 to 90% of transcripts, but there are some transcripts that are normally spliced. So although this variant does reduce the enzyme activity for the GAA protein, there is some residual enzyme activity, but it's been shown to reduce enzyme activity in cells and also in homozygous patients. So here we're showing the effect at the RNA level and also at the level of the enzyme function. And when I looked in ClinVar, I think there were 10 different labs that had classified this as pathogenic. And also of interest, when you look at adult onset patients, 36 to 90% of those patients in different studies actually have a copy of this particular variant. So that was one case, or one variant that was found in a patient. And I will note here, when I looked in exact, there was actually one homozygous patient in there. So I think this just really illustrates, we know from the data that's been published on Pompe disease that individuals who are homozygous for this variant, because it is a milder variant, because it can result in some residual enzyme activity, it tends to have later onset or very mild features. So it is possible that that person, you know, we don't know how old they were, this person who was found in EXAC. Maybe they just haven't developed features yet. Maybe they have extremely mild phenotype. But this did not make me think, oh gosh, this is not a pathogenic variant. It just made me realize that, you know, you really have to think about this information in the context of the disease. The second change that was found inherited from the father is actually a deletion of the whole of exon 18, but it is an in-frame deletion. And so when you look on Western blots of protein from fibroblasts from these patients, you actually see a reduction in size of the protein that is consistent with the deletion here. Individuals who are homozygous for this variant have no GAA activity. And again, this is a very common allele. It's found in about, has an allele frequency of 0.2 in Dutch patients with Pompe disease. So because these are common variants and there was a lot of evidence for them, I only this was the only case that I counted with these particular variants because I didn't want to kind of overinflate the score. And it's a definitive gene. I was trying to find a range of different variants for this gene. But I would give this case maximum points because of all of this evidence. But I know Marina and I had been talking a little bit about, these kind of situations where there are, common variants and really sort of how to handle that. Should you be counting these more than once? Is that inflating the score? Yeah, I mean, you certainly have to sort of think about the impact of what you count on your entire curation. And Marina, did you have any other thoughts on that?
I kind of just what I had said in the email. If there are one or two very common variants in a certain disease, unless those are the only variants, we try to score a range of different variants just to not give those variants too many points. But I know that there are many situations where there's really only, it's a definitive gene-disease association, and there's really only one or two variants, so it's understandable. And also the consideration of if you're going to count a variant multiple times, it's best to be fairly certain that the families aren't related of the two probands if you're going to count the same variant, because they could be distantly related, and that's essentially double counting.
Yeah, and part of the same family. And that's the thing, I think with these cases, a lot of these individuals do have a common haplotype, suggesting there's a common ancestor. So in some ways, they're almost part of the whole, a giant family in a way. So, yeah.
This is Courtney. I kind of had a comment or question on that, and I know you have to be careful, but is this kind of one of the things where the authors are kind of darned if they do tell us that they're related as opposed to if they didn't? Because, I mean, we've had papers where they'll first gene disease association and you'll have 10 probands and 6 of them have the same mutation, but you just don't know if they're related or not. Would you only count that variant one time instead of the other 5 times with the 5 different probands?
Yeah, that's a really good...
This happens to us a lot because we have, hearing loss, highly consanguineous populations. If we're really uncertain, we often turn to the experts to see what they think about it because it does happen so frequently. But if it's unspecified in the paper, sometimes we will downgrade the variant points if it's a highly consanguineous population. But I think it is a case-by-case because you don't want to inflate too much, like you said.
Right, and sometimes I worry just because, they're not giving us the information. How much can I infer that these 6 people, I mean, obviously they could be told like they're from different backgrounds if you're lucky enough with ethnicity, but even then, I think in the population today, I even had this question with Jen the other day where I had an exact score and the frequency for the Euro non-Fen was not somewhere we would count it, but the overall score was. And I'm like, how can I really trust when they say that they're Caucasian, that they're just Caucasian and European, and that they're not something else because you didn't really look through what their actual ethnicity was.
Yeah, I completely agree. It's best to be conservative and not assume anything if they're not telling you.